A eukaryotic gene is composed of upstream sequences such as the promoter, the RNA coding region, and downstream sequences. Many RNA coding regions consist of coding regions called exons and non-coding regions called introns. Many genes also contain a 3' consensus sequence. Before an RNA transcript is translated into protein, its pre-mRNA form is extensively processed. The first processing step is the addition of a cap at the 5' end of the pre-mRNA. Capping takes place rapidly after the initiation of transcription. RNA processing continues with polyadenylation. First, the pre-mRNA must be cleaved shortly after the 3' consensus sequence. Then, from 50 to 250 adenine nucleotides are added to the cleavage site. The next step in RNA processing is the removal of introns. An intron contains 3 consensus sequences critical to splicing: a 5' splice site, a branch point, and a 3' splice site. In the first step of splicing, the 5' end of the intron is simultaneously removed from the upstream exon and attached to the branch point. In the second step, the 5' end of the downstream exon is removed from the intron as the exon is spliced to the upstream exon. Splicing continues until all introns are removed. The mature processed transcript is now called messenger RNA or mRNA and contains the 5' untranslated region, the protein coding region, and the 3' untranslated region. The mature and correctly processed mRNA is transported out of the nucleus and into the cytoplasm, where translation by a ribosome can take place. The failure of any of these steps of pre-mRNA processing has consequences for gene expression. For example, if 5' capping of pre-mRNA does not occur, then the splicing stability and the translation of the RNA are all affected. First, the lack of a 5' cap will affect the removal of introns. Second, mRNA that lacks a 5' cap is less stable and will quickly degrade compared with mRNA that has a 5' cap. Finally, if mRNA lacking a 5' cap is transported into the cytoplasm, the ribosome will not recognize the mRNA, and the mRNA will then degrade. If splicing fails, 5' capping and polyadenylation will still proceed normally. If splicing is prevented by, for example, mutations at the branch points, then the RNA will be unable to move out of the nucleus into the cytoplasm and will therefore not be translated. If the 3' consensus sequence does not function properly, the cleavage of the pre-mRNA cannot take place. The result is mRNA lacking a poly-A tail. Splicing will take place normally in mRNA lacking a poly-A tail. However, an mRNA that lacks a poly-A tail is unstable and quickly degraded by cellular enzymes. Therefore, it will not be available for translation.